Congratulations on purchasing the Experion system. I'd like to take this opportunity to walk you through this short training video. The Experion system applies Bio-Rad's decades of knowledge and experience in protein and nucleic acid separations to lab chip microfluidic technology. The chips consist of a plastic caddy with a small glass plate affixed to it. The glass plate is etched with numerous microchannels and an optimized network pattern. The electrophoresis station directs the samples through these microchannels by controlling the voltage and current that is applied throughout the network of channels. By performing this procedure on a microfluidic chip, it is possible to accomplish several sequential procedures without any user intervention. The process of loading and running the chip is straightforward. Because the pipetting technique into a chip is unique to this method, however, it may take running a few chips to develop a routine process. This video will ensure you get your experiments underway by learning the proper pipetting technique quickly and properly. Loading liquid into the chip wells can be challenging the first couple of times, but will get easier with each try. Here are a few things to keep in mind during this process. You should make sure all your reagents are completely brought to room temperature for at least 20 minutes before proceeding. It is also critical that all tubes of reagents be inverted several times and vortexed to ensure reincorporation of any condensation formed upon storage. Then the reagent should be briefly centrifuged to collect the solutions at the bottom of the tubes. Make certain that you do not use coated or treated tubes, for example, siliconized polypropylene, during your sample preparation. When priming your chip, make certain it is seated firmly and flat on the priming station platform. You should have a successful priming every time, which can be observed by viewing the back of the chip and noticing that all the microchannels are bubble-free and fully filled with gel solution. Carefully pipette your sample using a calibrated pipetter and tips that are tapered. Hold the pipetter straight up and down. Always insert the pipette tip until it lightly touches the bottom of the chip well when dispensing liquids. This pertains to protein samples, gel and gel stain solutions, and RNA loading buffers, but not to RNA samples. For RNA samples, the tip should be positioned in the middle of the loading buffer present in the well and then dispensed to the first stop on the pipetter. Slowly dispense the liquid, taking care to not expel air. After all samples have been pipetted into the chip, you can visually inspect for air bubbles in each well by holding the chip over a piece of white paper. It is very important to make sure there are no bubbles trapped at the bottom of the wells before proceeding. The sample molecules move in the chip by the current and voltages applied throughout the platinum electrode pins in the electrophoresis station. Bubbles at the bottom of wells will prevent the completion of the circuit and will affect proper chip performance by causing IV check failures or late and odd electrophoretic behavior of sample molecules due to their experiencing of reduced electric fields. For the chip to function properly, each well must make good electrical contact with the appropriate channel. One way to observe bubbles is by visually inspecting the inside of the wells. A well with a bubble at the bottom will have a smaller circle inside than one that is free of bubbles. Here is what the chip looks like after the wells have been properly filled with solution. Now for comparison, here's what a chip looks like with several large bubbles present. Small bubbles toward the surface of the sample or gel solutions are not harmful to the run and will not affect chip performance. If you do find you have several small bubbles at the surface of the well, you can proceed without a run problem. Again, here is what the chip looks like after the wells have been properly filled with solution. And for comparison, here is what it looks like when small surface bubbles are present. If you find that you have a bubble located at the bottom of the well, it is easy to simply remove all the liquid in the affected well and replace it with a fresh sample for the protein assay. For the RNA assay, make sure you remove liquid after vortexing, then re-add loading buffer and sample and vortex again. You should make certain all wells are filled with liquid. If any of the wells are empty, the chip will not run properly. Once your chip is properly loaded, you are ready to place it in the electrophoresis station and begin your run. Within 10 minutes, you will already start collecting data on your first sample. In addition to faster results, the Experion user will benefit from reduced sample and reagent usage, automatic calculations, reduced hands-on time, and elimination of the challenges, tedium, and hazardous materials typically associated with traditional gel electrophoresis. By automating sample separation and analysis, valuable time is freed up, leading to faster research results overall. We are sure that with these simple guidelines, using the Experion system will be an easy, streamlined process. Thank you for your time. For additional information, call 1-800-4-Bio-Rad or contact your local sales representative. Have a great day.